Hello and welcome to the Light-Sheet Chronicles. I'm Dr. Elisabeth Kugler, a science communicator and your host of today's episode from stopping hearts to FCCS, using lasers to study tissue with light-sheet microscopy. In microscopy, lasers aren't limited to visualizing tissues with fluorescence, but scientists can manipulate and reshape lasers to gain new insights, for example, on cellular behavior and tissue dynamics. Today, we will talk about the things we can do with lasers, including how to use a light sheet to improve image quality, adopt application-specific beams, and use lasers to manipulate tissues. Our first guest today is Professor Dr. Jonathan Taylor from the University of Glasgow, who works on experimental imaging in optics, combining it with rigorous mathematical theory. And one of the research fields he is best known for is his work on synchronizing imaging with motion, such as when imaging the beating heart of zebrafish. Welcome, Johnny.
Hi, it's great to be here.
Thank you so much for joining us. And with us today as well, from Bruker Luxembourg, the Light-Sheet division of Bruker, we have Dr. Malte Wachsmuth, the Managing Director of the Luxembourg GmbH. So welcome, Malte.
Hello, thank you for having me.
It's fabulous to have you both here today, and we're going to talk about loads of fantastic things. And the first one we're going to talk about is actually how we can use the light sheet to improve image quality. So we know that in most imaging techniques, there is unfortunately some forms of artifacts, and artifacts can obscure details and reduce the accuracy, especially if you want to do quantitative analysis. So Malte, can you tell us about the different strategies that exist to address artifacts?
Yeah, sure. I mean, a major difference of light sheet microscopy compared to more conventional epifluorescence or confocal microscopy is that you're actually illuminating your sample from the side and perpendicular to your viewing direction. That means everything that casts a shadow shows up quite clearly in the image immediately. So you have these very, very typical striping effects in light sheet images that everybody who has looked at light sheet data probably knows quite well. And to overcome this, there are different strategies to mitigate this. One would be to illuminate not only from one side, but also to illuminate your sample from opposing directions, from the left side and from the right side, so that you can basically recover the signal in areas where the light from the other side casts a shadow. Or more generally, you can generate illumination from a variety of different angles so that you have a very similar effect. One way to do this would be to have multiple objective lenses that allow to illuminate the sample generically under different static angles. Another way would be to do this dynamically. What you can do, you can basically pivot your beam rapidly around the center, around the beam waist of the beam, which is typically in the center of the image, of the field of view. And like this, you can also generate a broad range of angles to illuminate your sample, which allows to remove these shadowing effects. So you can do it statically and also dynamically. Another less obvious and maybe a little bit more hidden artifact is that in light sheet microscopy, the illumination is not homogeneous across the field of view. You're illuminating, like I said already, you're illuminating from the side, typically in most cases with a Gaussian beam. And a Gaussian beam, the properties of a Gaussian beam are such that you have a beam waist where intensity is the highest and also the thickness of the beam is the thinnest, but it's converging towards the objective lens and diverging away from the objective lens so that you have really a inhomogeneous distribution of intensity and beam thickness. One way to overcome this would be to dynamically scan the beam waist axially back and forth. This can be done, for example, by introducing a tunable lens. That is something we like to do in our system, that we in an image plane that is conjugated to the back aperture of the objective lens, we introduce a very rapidly tunable lens that oscillates between negative and positive refractive power. With this, you can, like I said, rapidly axially scan the beam waist from the left to the right in the field of view, so that you have a very homogeneous, averaged out intensity and also beam width. Another way to solve this problem would be post-acquisition, by acquiring reference images where you can clearly see with a homogeneous sample, for example, of a dye in solution. Take these reference images and use this as a calibration image that you can then use to correct your actual image from an actual sample post-acquisition. This helps a lot with the intensity, but not so much with the beam width or the thickness of the beam at different areas across the field of view. And these are things one can address, for example, with calibrated imaging using methods like fluorescence correlation spectroscopy. If you have some time later in the course of our discussion here, maybe we can discuss this as well.
Yeah, and I'm so excited. We will make sure we get to this bit, which is the juicy science bit. So we're very excited about this. But you already brought in so many different things, such as image quality in general and artifacts, how they relate to each other. And you also mentioned Gaussian beams. And so maybe, Johnny, you can add to this and talk with us about how different beam types, such as Bessel and Airy beams, influence the balance between image resolution, penetration depth, and photo damage in light sheet microscopy.
Yes, so Malte alluded to this what's basically a fundamental limit of physics for a Gaussian beam that says there's this trade-off between, in the context of light sheet microscopy, the Z resolution that we can achieve versus the field of view over which we can achieve that because our light sheet remains thin and well behaved as we want it to. So people have tried there's all sorts of different variants of light sheet microscopy using different beam types people have tried to do, particularly ones that are referred to as non-diffracting beams, with the thinking being that these particular beam types will propagate for longer distances without having that diffractive expansion that loses the quality of the sheet. So this lets us influence a balance between the imaging resolution, the penetrating depth, and also the photo damage. The problem, though, is that this comes with a bit of a trade-off. So particularly with Airy and Bessel light sheets, you get these side lobes. So you don't just have a single thin light sheet, you get additional sheets alongside it. So you've got this trade-off, and I think this is quite interesting that it's well accepted that there's this fundamental limit of physics for a Gaussian beam, but I'm not convinced that, or I'm not aware of a really well-established theory that says that there isn't out there somewhere some kind of strategy for forming a light sheet that would have better performance than a Gaussian. And so people have spent a long time trying to find loopholes in this and trying to find a solution. But it does really seem like there is no free lunch. There's always a bit of a trade-off one way or the other. Although, as I say, I don't think there's any fundamental law that says it has to be that way. But so what that means in practice is that you can get an expanded field of view, which is great for getting high quality imaging over a wider field of view. But often, in order to get really good quality in terms of Z resolution, you then have to apply deconvolution to sort of undo the effects, or sort of take into account the effects of these additional side lobes. So your imaging acquisition pipeline becomes more complicated, but at the end of it, you do get better Z resolution over a wider field of view.
Lovely. Thank you so much. Our next episode is about image processing. So we look really forward to also talk about deconvolution and things there. But that was a great first insight. Thank you so much. I think one of the really interesting things that biologists really want to know as well is how we can use lasers to manipulate tissues. Because imaging a moving heart is incredibly challenging. And I think everyone who had a live sample under their microscope will know how challenging this is. And one approach to computationally freeze the heart for 3D imaging is achieved by what is called gated imaging. And that is quite commonly used in the clinical imaging, such as MRI, but is significantly less so used in the life sciences and preclinical models. But there was a study by Johnny and your colleagues that was published in 2019 in Nature Communications. And I remember reading that study during my PhD, and it was absolutely amazing and groundbreaking. And so maybe you could share with us what it really was that you did in that study and why it was so groundbreaking at the time.
Yeah, so like you say, I mean, a key challenge in biology is to separate cause from effect and understand what's happening through the process and not just imaging the final result. And of course, this ability to understand the time domain is the whole point of doing live imaging, such as with light sheet microscopy, seeing how things change over time to better understand the causality and the mechanisms behind what's happening. But then for samples that are constantly moving, with the heart being the extreme example of that, you've got the problem that you're wanting to capture a series of images and understand the sort of the biological changes that are happening on a time scale of minutes to hours in that tissue, whether that's cell division, cell migration, changes of state of the cells. But then if you're completely overwhelmed by the large beating motion of the heart several times a second in the case of the zebrafish, you're not able to see the structures that you're actually wanting and the phenomena that you're wanting to understand. So we had to develop our technical approach to computationally freeze that motion to enable us to see stable three-dimensional images over time. And so the way we were able to do that was to image continuously in a bright field channel and monitor in real time the motion of the heart, which enabled us to then make real-time decisions as to exactly when to trigger individual fluorescence images to give us a stable image, as if the heart wasn't beating at all in the fluorescence channel. But of course, we know that it is constantly beating. It's just through our sort of stroboscopic timing of when we capture the fluorescence that we were able to get that stable image and that series of stable images over time.
Yeah, and I think it's also important to note that zebrafish that you presented in that study have a very rapid heartbeat, right? Around 120 to 180 beats per minute. So the whole system and all the computation needs to be very rapid, right?
Yeah, we're having to track the rhythm of the heart over time, because it wouldn't be enough just to assume that the heart is beating at a constant steady rate. So we're having to process images at about 100 frames a second, make real-time decisions from those, and then determine when that upcoming feature correct time will be to capture the next image.
And Malte, do you think that it is the same with like optogenetics, that you really should kind of stay on track with like the movements, the biological timeframes? Because again, we use optogenetics for slow but also quite fast processes.
I think, yes, we should do everything we can do to also get a comprehensive view on your sample and on your object from the very beginning. And then after you really reach the limits of what's technically possible in terms of speed, in terms of acquisition, resolution in space and time, and then move over to additional tricks like optogenetics or your gated imaging. I think you should really push things as far as possible with full speed acquisition in the first place, and then put additional technologies on top.
There's so many interesting approaches, and I think especially also as a biologist, it's really exciting to see that we can use lasers in so many different, ways, and also like the actual computation to really get the most out of our data. And one thing that we really wanted to discuss in this episode is also really more advanced techniques, because I think especially in the last few years, we've made huge steps to using, for example, FRAP in many different labs. And so I thought it was really important that we discuss things like this today. And so because we know FRAP, which is fluorescence recovery after photobleaching, we thought today we're going to take all of this a step further and actually kind of skip FRAP a little bit and directly dive into FCCS, because FCCS is fluorescence cross-correlation spectroscopy. And this is something that is significantly less used in many labs and also probably less known. So Malte, can you maybe speak to our listeners, what is FCCS and why it is so amazing to have in different labs?
The FCCS stands for fluorescence cross-correlation spectroscopy. And maybe let's step back a little bit and start with FCS, which is fluorescence correlation spectroscopy. That's a method that has been termed an emerging method since 1970 or 1972, but it's really coming of age finally. Not least because it's being used on the platform of Light-Sheet and also TURF systems, so camera-based system basically. And the major challenge of FCS is that it's a point spectroscopy method. So it's originally based on a confocal setup, like you know probably from a confocal microscope. So you park your beam at a spot inside. Initially, it was mostly used as an in vitro analysis technique, but this is as powerful in vivo, inside living cells and living tissue as it is only in solution. And so you park your beam at a spot, and then you observe molecules as they pass by diffusion or also flow. And every molecule that enters your focal volume and leaves it again. It generates a kind of like a flash of fluorescence photons, and the length of these flashes or these fluctuations are in average represent the mean dwell time of your molecule inside your focal volume. And from this mean dwell time, you can learn a lot about the size of the molecule, its molecular weight, and also its binding status. Think of a small molecule that binds to some extent to a larger complex. Then obviously the small molecule that is somehow fluorescently labeled has a much shorter dwell time as a free molecule, and a much longer one when complexed to its larger binding partner. And a way to extract this mean dwell time from these intensity traces you measure from a single spot is a so-called autocorrelation analysis of the signal. That means you compare basically the signal with itself, and you shift it, and you basically make a copy of your data set, and you shift it in time, and then you get an overlap of these fluctuations that decay over time. Once it's very unlikely that a molecule has stayed that long inside the focal volume. And from this, you can determine the mean dwell time, and also from the amplitude of your so-called autocorrelation function, the number of molecules in your focal volume. And once you know the volume itself, you can also determine a concentration. This takes me back to what I said before, that you can use this to also calibrate a fluorescence image. It's something we've done a lot in earlier days with confocal imaging. So you can basically then say, okay, I can measure a concentration at a spot, also have an intensity value at the spot, so I know to which concentration a certain intensity value corresponds. Like this, you can calibrate your intensity images, and you can also correct for these inhomogeneities of illumination that we've discussed before in light sheet microscopy. So in a way, it's when you then basically are interested in the interaction of two types of molecules that carry different spectral labels, then you speak more about a kind of like a dynamic co-localization of molecules. So when these two little guys bind to each other, then they co-diffuse, and then they create synchronized fluctuations on two spectral detection channels. And when they do not bind, then they basically show independent fluctuations. So when you then determine a cross correlation of these two intensity traces in your two spectral channels, you can determine the amount of interaction. And after all, especially with knowledge about the concentration, you can also determine things like a dissociation constant. So really quantitative biochemical parameters. And so far, I only mentioned that you can do this on a confocal system, but the big plus of light sheet microscopy is that you don't have a single confocal volume, but you have a two-dimensional array of confocal volumes. So when you can do this in an imaging way, meaning on every single voxel of your camera array, then you can also turn this into an imaging method. And this is basically what light sheet microscopy has made possible by using highly sensitive cameras as CMOS cameras, and what's emerging now is also real photon counting technology for two-dimensional camera arrays. And with these tools, it's possible to generate concentration maps, interaction maps, and potentially also KD maps of interaction partners inside of a living cell, inside of living tissues.
Thank you so much for that wonderful summary, Malte. As a biologist, I know there's so much kind of maths and physics involved. And I know I've done myself FCCS with you for my master thesis many, many moons ago. And I remember we just needed a lot of prior knowledge as well, right? About dwell times, about fluorophore sizes, migration times, etc. So that was lots of different things that we needed to really involve. But the great thing is, as you say, what we can then do and find out is really biochemistry in a living sample without actually being invasive. And so, for example, as you said, we can measure concentrations, but also binding or mobility or co-localization of, for example, proteins, which is really amazing. I think, so like I said, one really difficult thing is that really the implementation is quite challenging, especially getting all the maths right and setting out the experiments in the correct fashion. But I assume, as with most techniques, applying it from confocal to light sheet is really a very different challenge. So maybe you can talk with us about what makes it even more challenging to integrate a technique like FCS or FCCS with light sheet microscopy, Malte.
Yeah, completely right. The challenge doesn't necessarily lie in the acquisition process, but it's really the processing that is the most challenging part of this kind of experiments. The good news is that nowadays you don't have to take care of this yourself anymore nowadays. So you can, for example, from the lab of Thorsten Wohland at National University in Singapore, he has developed a very, very nice Fiji plugin for the acquisition, processing, and also interpretation of FCS and camera-based FCS and FCCS data. So the correlation functions are calculated automatically with GPU accelerated algorithms, and also the fitting is not necessarily traditional curve fitting anymore, but they use a lot of machine learning, and it's quite amazing how well this works when you compare this to ground truths based on fitting of data. So this has gotten significantly more robust than what we know from 10, 15 years ago. And I think this really turns it into a tool that is actually usable also outside a niche of biophysicists who know a lot about mathematics.
I have not heard that you said my master's was like 10, 15 years ago. No, yes, absolutely. And I think that is the beautiful thing, that bioimage analysis is really supporting the imaging in such a beautiful way now. Like you say, GPU accelerated computation, but also machine learning, AI, event-driven microscopy. There's loads of really interesting stuff happening. So I'm really excited also about the next episode of the Light-Sheet Chronicles. As I mentioned, we're going to talk about image analysis. So lots of really great science happening there as well. But now to switch gears a little bit, on the other hand, to FCS and FCCS, there is a very different technique, which is FLIM, so fluorescence lifetime imaging. And Johnny, I know it is really becoming a technique that is very interesting to the community. So maybe you can talk with us a little bit in general. What is FLIM and why is it so interesting at the moment?
So it's tackling some of the same challenges that Malte was alluding to, but by a slightly different method. So for fluorescence lifetime imaging, so whenever we excite fluorescence using a laser, most of the time we assume that the light we get back is basically instantaneous. But in reality, we excite the laser and then there will be a stochastic delay of we excite the fluorophore with the laser, and there will be a stochastic delay of a few nanoseconds before a photon is actually emitted. Most of the time that doesn't matter. But that sort of average delay is the fluorescence lifetime. And if we can measure that, if we've got precise enough imaging hardware and timing electronics to measure that delay, then we find that different fluorophores have all got slightly different fluorescence lifetimes. So it's already a different mechanism for contrast. But that lifetime is really sensitive to the environment that the fluorescent molecule is in. So it's very sensitive to pH, temperature, and interestingly, proximity to other fluorophores. And so you're starting to get a biochemical insight into the tissue that you're imaging, rather than just structural information. You're starting to learn stuff about the environment that molecule is in. So it requires relatively expensive hardware to be able to do those measurements. But if you can do that, then you're learning a whole lot more about, in my case that I'm particularly interested in, the living tissue that you're imaging that you couldn't image just through structural imaging.
Yeah, and I also know that you're actually taking all of this to a completely new level. So you're not just using FLIM, but also FLIM-FRET tension sensors, which is like really next level imaging. Can you talk with us about what you're doing there and what is so exciting about it?
Yes, so these tension sensors are fantastic. So this is really sort of building on this concept of proximity influence between different fluorophores. So Martin Schwartz's lab have developed these absolutely wonderfully physical tension sensors where they've basically taken a molecular linker protein, but they've embedded into it two different fluorophores that are separated by a short length of elastic spider silk peptide. So this has basically made two fluorophores attached by a really tiny molecular spring. And so then the FRET process means that the fluorescence lifetime of one of these fluorophores will be modified depending on how close the other one is to it. And so you've basically got a readout of fluorescence lifetime where the lifetime changes depending on how much this elastic linker is stretched, which in turn is telling you about how much tension that particular molecule is under. And then Anna Lagendijk engineered this into zebrafish cadherin. So there was a live zebrafish model that embedded this into the endothelial cells of the fish. And so you're getting this molecular tension readout in a live animal. But then you've got the additional challenge of doing FLIM-FRET in a much messier in vivo environment with a lot of other background fluorescence in there as well. And then in our case, where we want to do this sort of in the hardest place possible, we want to do this in the heart, you've got the additional problem of the moving tissue. And the reason that we want to be able to do this is that knowing what these intercellular forces are is absolutely crucial for morphogenesis, both in terms of the mechanical forces that the cells are under in itself, but also how the cells are responding to those forces, as well as just basic questions about the heart function and how those cells are interacting collectively together and the force that they're generating spontaneously. And so because we're in the heart here, we're back to this problem of imaging moving tissue. We want to be able to image this in a wide field snapshot because we want to be able to see, the whole field of view of the heart. And then we've got the additional challenge on top of all of this that FLIM-FRET is typically a very photon-starved modality. You're really struggling to get as many photons as you possibly can out from these fluorophores to be able to make these measurements, which again connects in with your next topic of the image analysis and the challenges of working with very low photon count images. So our current research focus is on developing technology to solve all these problems, to be able to do FLIM-FRET in vivo in the beating heart. And that's really needed us to get together a multidisciplinary team that can work together on the probes, the optics, the imaging technology, and the downstream data analysis, bringing all that together to be able to image molecular forces in context across a complex functioning organ such as the heart.
I'm listening to you and I'm just nodding and I'm like, oh my God, your work is so amazing. It's really, really interesting because especially you build on work, like you say, that has been partially published before, but you take it to a completely different level, especially in the heart is so, so challenging. And tension sensors, oh my God, love it. Very amazing work. One question I did have on that side question is, so basically with the heart, beating heart being obviously kind of distributing the forces to the surrounding blood vessels, right? And we know that, for example, the Windkessel effect can be really impactful, especially around the gills, the brain. Could that be something where you use these tension sensors as well? So you see kind of the tension that the tissue just around the heart is kind of being impacted on?
I think there's a huge amount to explore, and I think we're starting out with just one organ, but because at the moment we're quite limited in our field of view. A whole other story about the imaging hardware, the imaging hardware cameras to be able to make these measurements in a wide field manner, where traditionally it was done in a point scanning regime, means that we're a little bit limited in field of view at the moment. But yes, I think that question of how this all integrates across different organs and different regions of the fish in our case is a really interesting question to explore. I think what I find so interesting and exciting about in vivo imaging is also the thing that makes it really hard and challenging is that if you were trying to ask a simple question that could be boiled down to something that could be studied just in cultured cells or in an explant or a simple organoid or something, then we wouldn't be trying to do this in vivo because there would be an easier solution. But when you're tackling these complex questions, you really need to know what is the whole system, what is the whole complex living organism doing, and all of that is feeding into the biological question, which means that all of that has to be taken into account and included in the imaging that we're doing, ideally.
Absolutely, I couldn't agree more. We've spoken about so many different things, but there's obviously also so much research happening. So Malte, we wanted to ask, what is the one thing that you are the most excited about the future in light sheet microscopy?
Oh, the one thing that's really difficult to answer. I think the one thing that I'm most excited about is really the broad range of applications you can address with light sheet microscopy. So we talked about imaging basically on a single molecule level by looking at cross correlations of co-diffusing molecules at the lifetime of individual molecules that can sense tension via a FRET effect. On the other hand, you can look at an entire cleared mouse, or you can look at expanded tissue to have kind of super resolution like impact. I think the one thing that I find most fascinating is really the versatility. This keeps growing and keeps being a challenge.
Absolutely. And Johnny, what about you? Is it the tension sensors?
So on a specific level, absolutely. I'm super excited about the tension sensors and the potential for using those as an in vivo probe. But I think more generally, along the lines of what Malte was saying, for the field as a whole, what I think is really excited about where we are now with the field of light sheet microscopy is you don't need to be an optics expert to use these systems and to benefit from them. For many years, when I was starting out in this field myself as a postdoc, the light sheet, there was an annual light sheet fluorescence microscopy conference, which was absolutely fantastic to go to. Really, really excited, really collegiate field focused on technical development as well as applications, and a lot of that was around home build systems. But now, I miss that community excitement and that home build atmosphere. But actually, I think it's a great sign of a mature technology, almost on a par with confocal, that this has been largely replaced by user workshops of biologists who are applying this tool to answer their questions sort of on their own, if you like. And so, I'm excited for the technological, but also the biological applications that are coming out from what's now a very mature field.
Thank you so much, both of you, for these wonderful insights. It was great to have you. Thank you, Malte and Johnny, for both of your time today.
Thank you very much.
Thanks.
But unfortunately, this is all that we have time for today. And so we talked about the things we can do with light sheet lasers, including how to influence artifacts, adopt application-specific beams, and use lasers to manipulate tissues. Huge thanks to our listeners for tuning into this episode of the Light-Sheet Chronicles. And in our next episode, Image Analysis in Light Sheet Microscopy, we talk about big data processing, handling, and storage. Thank you and goodbye.
This podcast was sponsored by Bruker Luxendo GmbH, the light sheet division of Bruker's Fluorescence Microscopy division. Bruker is a manufacturer of scientific instruments for life sciences, materials research, and industrial or applied analysis. If you'd like to learn more, please visit www.bruker.com/light-sheet. This podcast was produced by Keep It Science.